Good morning, everyone, and welcome to Arthritis at Home. Today, I'm joined by Dr. Landon Edgar, who's an assistant professor in the Department of Pharmacology and Toxicology, as well as the Department of Chemistry at the University of Toronto. More recently, his team was awarded the Ignite Innovation Grant from the Arthritis Society for their project titled Tracking and Stopping Spine Arthritis Using Carbohydrates on Immune Cells. Very, very cool title. And what I'd love to speak to Dr. Edgar about today is this exact project, and we will hopefully learn a little bit about what are carbohydrates and what are immune cells. So with that, thank you so much, Dr. Edgar, for joining us.
Thank you so much for inviting me to speak here today. I'm really excited to have this conversation.
Wonderful. So Dr. Edgar, can you tell us a little bit more about yourself and how you're involved in rheumatology?
That's a great question. So I took a rather unconventional pathway towards arthritis research. So my doctoral work was in pure synthetic organic chemistry, where I spent a lot of time mixing chemicals together to make organic molecules that we eventually use to study some tumor biology. So pretty separated from arthritis research. And at the end of my doctoral degree, my PhD advisor strongly recommended that I go off and learn some biology somewhere else. So I took his advice, which was great advice, and went to the Scripps Research Institute in La Jolla, California, to do what's called post-doctoral research, which is an additional set of training after your PhD so that you can learn new skills and sort of enter a new field where maybe you can make a unique impact by combining your doctoral training with a question, a new question in a postdoc environment. So I did that work in glycoimmunology. And what that means is the study of how the immune system is dependent on glycans, or complex carbohydrate structures that exist on literally every cell in every organism, including humans. So after engaging in that postdoctoral research, I was interested in an academic career. So I thought, what could I sort of uniquely offer to the field as a synthetic chemist with this glycoimmunology training? And this was actually right around the onset of COVID that I was on the academic job market, and was thinking about vaccination and immune responses. And something that had struck me was that we're really good as a society or as scientists at initiating specific immune responses. So we can do that through vaccination. We do it for influenza, we do it for SARS-CoV-2, we do it for measles, mumps, rubella. What we're not very good at is turning off specific immune responses. So doing exactly the opposite of what a vaccine is designed to do. And that got me thinking about, well, if we could do that for autoimmune diseases, that would be probably the best possible treatment, because an autoimmune disease, by definition, is really an unwanted immune activation or an immune response. Arthritis is a great example of that. So during my postdoc, I was part of a team that discovered that carbohydrates on immune cells, which we'll talk about later, really play a major role in tuning how those immune cells respond to specific stimuli. And so I thought maybe that's something worth exploring from an immunosuppressive angle. And then I was fortunate to get hired at the University of Toronto in my current position, and thought arthritis would be a fantastic jumping board to test some of these hypotheses. So that's really how I ended up here.
Wow, it's a beautiful story of not only, finding your niche as a researcher and academic, and then knowing what is kind of the next innovation, right? And what to jump on board. So I'm so grateful to have you in the field. And as a patient myself, I'm like, I cannot wait for what's to come of your research.
Thank you so much.
So Dr. Edgar, tell us about, in lay terms, what is an immune cell? Because of COVID, we hear all of these long scripted names of what an immune cell is, but basically, what is an immune cell? And like you said, a carbohydrate, and I think you said you used complex, right? Complex carbohydrates. What are those?
All right, so I'll answer the first part first. So, what is an immune cell? Well, your immune system is effectively a whole constellation of different types of individual cells. And each of these types of cells has a specific function to keep you in what we would call homeostasis, which is a fancy way of saying, healthy and without disease. And so disease can manifest as many things, right? You could have a viral infection, or you could have a wound infected with bacteria, or maybe you have cancer or an autoimmune disease. In all of these cases, immune cells play a central role in either helping to bring you back to homeostasis, to a healthy state, or if there's dysregulation, keeping you in a state of unwanted inflammation, for example. So as I said, there are many types of immune cells. What we typically think about in my laboratory are specific ones called thymocytes or T cells. And these are immune cells that act in very specific ways against very specific biological matter that we call antigens. So SARS-CoV-2 has a lot of antigens on it. The most important one for therapeutic purposes is spike protein. So spike protein is a piece of biological matter from the virus that our immune system can recognize and specifically mount an attack against if it's being given that information, that antigen is a piece of biological information. And T cells we think a lot about, are really unique in that they specifically recognize that particular antigen and will only mount immune responses against that antigen and not touch anything else. Some of that is a lie, but for the purposes of today, I think that's good enough. And so that's a really powerful piece of information, right? If you can harness T cell immunity to specifically attack something that you want to, say, mount a response against, then you should be able to, say, clear infection. And that's what a lot of vaccines do. They give the T cells the instructions. But T cells actually play a major role in autoimmune diseases like multiple sclerosis, type 1 diabetes, and arthritis. Ankylosing spondylitis, rheumatoid arthritis have a major T cell component where your T cells are now recognizing a piece of you and attacking that piece of you. We call those autoantigens that they recognize. So shutting off those T cell responses against autoantigens is a really important topic in the field. To answer the second part of the question, what's a complex carbohydrate? Well, I mentioned earlier, every single one of the cells in your body, including your T cells and all of your immune cells, are coated in a dense, you could think of it like a forest of complex sugar molecules. And I'm not talking about the kind of sugar you put in your coffee or your tea. That's nutritional carbohydrate. These are immunogenic carbohydrates. They have very specific monosaccharide compositions. So there are building blocks that make these complex structures. You can think of it as almost like Lego. And they're displayed out on the surface of all of these cells for the rest of your body to see, including other immune cells. And the thing about sugars, or glycans as we call them in this context, is that they're not encoded directly in your genome, which means we can't just use sequencing technologies like DNA sequencing to figure out what carbohydrates are on your cells. You can't sequence them. So what that means is that glycans in carbohydrate biology, we call it glycobiology, has lagged far behind almost all other biological research. And that's because it's just really hard to look at these structures. So it turns out that most good glycobiologists are originally organic chemists by training, because in order to access the molecules to be able to study them, we can't use traditional molecular biology or biochemistry approaches. We have to go into a chemical laboratory and literally make the molecules, in some settings, atom by atom. So that slows progress a lot, but it really excites me because this is a wonderful discipline to be at that's really intersectional with so many different compartments of science.
I think it really does speak to like you were sharing in your story, right? Just again combining, what you had studied and you were passionate about with this to solve this really, really interesting question. So I think that's absolutely incredible. And your description of a T cell, right, as those defenders in our body, but not just any defender, a defender that can differentiate, which means it can be specific and knows who to attack and who is a loving, fellow body cell. And then the fact that, like you said, complex carbohydrates, I really needed that differentiation of complex carbohydrate versus dietary carbohydrate and knowing those two differences. So thank you for that. Next question for you is, how does these complex carbohydrates, how do they coat an immune cell, and what does that actually lead to for patients with axial spondyloarthritis?
Well, what does it lead to for patients with axial spondyloarthritis? That's the question that we're trying to answer right now. So we have lots of hypotheses that need to be tested, and really hugely grateful to the Arthritis Society for being courageous enough to risk putting money where these hypotheses are, because it's really cutting edge. We just don't know, but they're providing us with the means to go and look. And that's the first step to really any major discovery, is to see what's there. So that's what we're trying to learn right now. But going back, and I think you would ask, what does this carbohydrate coating sort of look like? What's the context of that? Well, these complex carbohydrates, these glycans, are tethered to the surface of your cell, usually by being literally attached through what we call covalent bonds to proteins that are on the surface of all of your cells. And sometimes they're attached to fat, to lipid molecules that make the membrane of all of your cells. And different immune cells in your body have completely different carbohydrate structures on their surface. So it's not just one type of complex carbohydrate. There's a whole forest, a whole repertoire of structurally very complex carbohydrates on the surface of all of these cells. And what we're trying to figure out is, how do we relate the specific carbohydrate structures to the specific immune cell activity? And now that we know that there is a relationship there, this was a major feature of my postdoc work, we found that certain carbohydrate structures on T cells actually suppress their activity. And if you go in and you artificially remove them, the whole wormhole of technologies to describe how we do that, but you can go in and selectively remove some of these structures. And when you do that, you greatly enhance the T cell's ability to mount an immune response. So that means that just the carbohydrates are tuning these very important immune responses on T cells. And this wasn't really appreciated, at least in a modern sense, until we started to do some of this work at Scripps during my postdoc. And so that's a new dimension to immunity. We've always thought, if you open an immunology textbook, you'll see a bunch of cartoons showing proteins on immune cell 1 and proteins on immune cell 2, and they come together and they signal, and that's how an immune response happens. But that's not the whole story. This dense forest of sugar is playing at least as important a role, it seems, as these protein-protein interactions. And now we're really poised to start to dissect some of the fundamental immunology here, and then look and see in arthritic patients if their sugar coating is different than a healthy individual. How does that look? And we have some reason to believe at this point that the sugar coatings are changed, and maybe that's a new therapeutic target in the future.
Like you said, incredibly cutting edge. And the fact that I didn't even know that there were already these complex carbohydrates that act, as you said, almost like a dimming switch, for your lights.
Yeah, exactly.
All right, defense team on. No, off. Guys, ease. Right. It's just this, it's incredible. Like you said, instead of a go or no go, it is truly like a spectrum of which they can be activated. So fine-tuning that, right, and understanding that process. Amazing. Can I ask, because this is so cutting edge, and like you had said, you are greatly at an advantage with your chemistry background, how do you actually look at carbohydrates coating immune cells again? Or maybe walk us through that step.
Oh boy, that's such a great question. You're asking just fabulous questions. How do we look at carbohydrates? Well, we actually rely a lot on, believe it or not, proteins made by plants. And these are taken from a plant, a specific species of plants, purified, And then they're usually labeled with a group that will respond to light that we shine on it in a specific way. We call those fluorophores. And these plant proteins, we call them lectins. Lectin is a fancy word for protein that binds to carbohydrate. We then take those and we incubate this plant lectin with the fluorescent group on it with our immune cells. And those lectins will only bind to immune cells that display a known carbohydrate pattern. And then what we do is we use some really cutting edge technology. It's called a spectral flow cytometer. My lab is fortunate to have our very own instrument, which we run almost 24/7 sometimes. And that instrument can analyze up to 20 or 30,000 immune cells per second and give us data in up to about 27 dimensions. And several of those dimensions can be just figuring out what type of immune cell it is. We use antibodies to do that. And the other dimensions can be to assign carbohydrates to those immune cells based on the lectin staining pattern. We also have collaborations where we can use a technique called mass spectrometry to go in and look at how much, for lack of a better term, what is the weight of some of these carbohydrates on the surface of the cells? And that is associated with particular structural features as well. So we use multiple techniques together to generate a more complete picture. But, for protein, it's a little easier. And for nucleic acids, it's even easier because you can just go and sequence things, which you just can't do for sugar. So if somebody listening can develop a carbohydrate or a glycan sequencing technology, there's probably a Nobel Prize with your name on it, but we're not there yet.
Well, you never know. That might be your project after this one. So I wouldn't put it past you yet. No, that's incredible. And for our audience, I'm going to use my understanding, and Dr. Edgar, let me know if I'm incorrect, but essentially you distill something from a plant. You take something from a plant, an extract from a plant, you throw it in there with all of the proteins that have the little complex carbohydrates on them, little dimmers on them. And the plant extract will only bind the dimmers. Then you shine a fancy light and you can see which ones are the appropriate ones for you to examine. Is that?
That's a great description of it. Yeah. And our flow cytometer is the thing that's shining the light on the cells.
Awesome. Awesome. Thank you so much. And Dr. Edgar, I do want to, congratulate you to lead this research. And I know there are many researchers on your team. Would you like to acknowledge anyone at this moment for their work?
For sure. So first, I'd like to acknowledge Dr. Robert Inman, whom is a clinical rheumatologist and physician scientist at Toronto Western Hospital. He's actually a co-applicant on this Ignite Innovation grant with me. And Dr. Inman really provides the deep ankylosing spondylitis expertise to this research team. I view myself very much as a basic scientist with some translational interests, but I'm not a clinician. Dr. Inman is, the rheumatologist on the program. So I think combined, we can achieve some really amazing things. His clinical perspective and his deep expertise in the immunology of arthritis, combined with a lot of our glycan detection platforms and technologies in my lab here. I'd also really like to thank some of the students who are working on this. Hani Chosky is an MSc student in my lab right now, and she's been driving this project. Melissa Lim in Dr. Inman's lab has been helping us with accessing the samples we need to measure some of these glycosylation changes in settings of arthritis. And we're really poised at this point, I think, to make some really meaningful progress in this space, which is just ripe for discovery right now.
And well chosen by the Arthritis Society. So we'll thank them again for their contributions to research. Thank you for everyone who donates to the Arthritis Society for allowing this work to be possible if you are watching. And with that, Dr. Edgar, want to say we appreciate your time. We appreciate you sharing your knowledge. And we really do wish you, your students, and Dr. Inman all the best in research.
Thank you so much for making the time to speak today. Again, thank you to the Arthritis Society as well for supporting this research. And I hope we get to speak again in the future where we're talking about maybe some therapies in the pipeline.
Can't wait. Looking forward to it. Thank you. And to our audience, have a great rest of your day. And we hope to see you on our next episode.
Take care. Thank you.